So remember, when we talk about pillar 1, you can replace the gene, you can replace the protein. Genes make protein, so the gene in a gene replacement therapy would be around hopefully forever, but the proteins that it's making are those that are being recycled. So your proteins would never be around forever because proteins get broken down naturally in the body. Our next speaker is Christopher Luthers. He's coming to us from University of UCLA, University of California, Los Angeles. He is going to be speaking to us about hematopoietic stem cell gene therapy. I told you HSC-GT, also pillar 1. Chris is actually a PhD student out of the laboratory of Dr. Don Cohn, who's a world expert in hematopoietic stem cell gene replacement therapy, and Chris is going to walk you through that a little bit and some of those details. So, Chris, come on up.
Hello, everyone. First of all, I'd like to, again, thank the organizers for inviting me to speak today. My name is Christopher Luthers. I'm a 5th-year molecular biology student at UCLA in the lab of Dr. Donald Cohn. And so today I'm excited to talk to you all about my research, hematopoietic stem cell gene therapy for the potential treatment of Angelman syndrome. So this is my disclosure. And so in my lab, we focus on ex vivo autologous stem cell gene therapy. It's a bit of a mouthful, but I hope to be able to walk you through each step of that. So if we look at the figure on the left, on the bottom, this gene therapy workflow is a cycle where we can basically collect the patient's bone marrow cells. From there, we can isolate the stem cells, the hematopoietic stem cells. So those are the stem cells from the blood or from the bone marrow, using this marker CD34. And then we can actually return the rest of the blood to the patient. So all we extract from the patient is their bone marrow stem cells. From there, we can modify the stem cells using a Lentiviral vector, which I'll explain a little bit more in the next slide. And this Lentiviral vector will contain our gene of interest. So in this case, it would have UBE3A. So we can use our Lentiviral vector to give our stem cells UBE3A, and we can take those stem cells and infuse those back into the patient. And so some of the benefits of this autologous gene therapy is that these hematopoietic stem cells, or HSCs, have long-term renewal potential. And so as Allison was mentioning, this basically means that if you're able to edit your stem cells, this would represent a one-time cure. So there would be no need for redosing or readministration. This gene therapy would need to happen one time, and then your stem cells would be expressing UBE3A for the rest of the patient's life. It uses the patient's own cells, so this overcomes the need for a bone marrow match donor. And so if you wanted to have a bone marrow transplant and you don't have a match, that's a limitation. And also if you have bone marrow transplants, there's the risk of your body rejecting the transplant, whereas if you're using your own cells, that's not a concern. And also ex vivo editing, so editing outside of the body is ex vivo editing. That prevents off-target editing of non-desired cell types. So we're only going to edit the CD34 cells, the stem cells that are outside of the patient. So what are lentiviral vectors and what are their benefits? So on the left here is a picture of a lentivirus. So a lentivirus is actually a modified form of the HIV virus that we all know that causes AIDS. But we remove all of the parts of the HIV virus that are dangerous. So the dangerous part of HIV is that it can replicate, so our vectors cannot replicate. The only thing they can do is have our gene of interest and integrate that gene of interest into one cell. It can't integrate into multiple cells, it can't make more of itself. And so the benefits of these lentiviral vectors is that they have low cell toxicity. They're easy to produce in labs, so we can produce large batches of different quantities of these lentiviral vectors. They enter our immune cells very efficiently, and they have a very good clinical safety profile. And so this is from an article in 2022. From 1995 to 2020, there have been 55 gene therapy trials with 406 lentiviral gene therapy patients, 99% survival rate of those patients, only a 1.4% incidence of toxicity, and 97% of patients show what's called engraftment of their lentiviral vectors. And this basically means that those stem cells were able to enter the patient's body and be producing, in this case, the UB3A protein for us, for the remainder of that patient's life. So those stem cells were able to engraft in the bone marrow and produce that protein, representing this one-time cure. So a little bit of details on how does this UBE3A addition work in stem cells. And so on the left here, you can see a schematic where we have our UBE3A gene in green. We can put that into our lentivirus. Then we give the lentivirus to our patient stem cells, and now we can create stem cells that express the UBE3A positive protein. And so if you look at the figure on the right, this is basically a schematic of the stem cells, which are on the top, all the way down to all the cells in between, which are these intermediate cells called progenitors, and our mature cells, which are on the bottom. So if we look at the stem cell and we see it in red, think of that as UBE3A negative. So if we're able to give the lentivirus to our stem cells, we can correct UBE3A in our stem cells, as indicated by the transition from red to green, and correct that stem cell to be expressing UBE3A. From there, the stem cells will make all of the mature cells in our immune system, switching them from red to green, basically allowing for UBE3A to be expressed across the entire immune system, which is what allows for this widespread UBE3A expression. But you're probably wondering, how can we use immune cells to cure a neurological disorder like Angelman syndrome, which we know is mainly expressed in the brain, or the limitation is mainly in the brain. And so this is this idea of cross-correction, where we can use gene transfer. So we can use our lentiviral vector, as I mentioned, to express the UBE3A protein. But what's key here is that UBE3A protein would then be secreted from our immune cells, uptaken by our recipient neurons, and the neurons can then use the UBE3A that the immune cells are creating. So again, the lentivirus would be infected into the immune cells to produce UBE3A to be uptaken by the neurons that can use it because the neurons are UBE3A deficient, and I'll show a little bit of that towards the end of my talk. So the work that the sort of paper that my work builds on is this paper from Joseph Anderson's group. And on the left, you can see two vectors. There's a lot going on, but if you just focus on two parts of the vector, the MNDU3, I'll highlight here. So the MNDU3 promoter. So promoters are what drive expression of genes. So you can have different strengths of promoters. And so MNDU3 is a very strong promoter, which makes the UBE3A protein. And so they showed that they're able to get expression of the UBE3A protein on the top right. And then they show a variety of corrections in the Angelman syndrome mouse model. But on the bottom right, I'm just showing one, which is the latency to cross a balance beam. But I'll summarize by saying this was a really promising result, and that they were able to show correction of the Angelman syndrome phenotype, or the Angelman syndrome mouse model, in a lot of different readouts. I'm just showing one here. But again, I want to highlight this MNDU3 promoter. So again, the promoter is what drives expression of the UBE3A gene, and MNDU3 is a very strong promoter. So strong that there was a press release in 2021 for another gene therapy trial using a vector that has that same MND promoter, in which one patient developed what's known as myelodysplastic syndrome. You can think of that as a precursor to cancer or precursor to leukemia, and another two had abnormal bone marrow cells that could lead to that myelodysplastic syndrome, so a precursor to a pre-leukemia, so early indications that they could develop cancer. And when scientists looked at their cells, they found that the lentiviral DNA had inserted into a site in the genome, which could lead to cancer. So it's the MND promoter and the strength of its promoter which is causing these cancers. And there was a paper that just came out about a month ago that was detailing some of these cancers in the gene therapy trial from the press release on the left. So 10% of patients had developed this leukemia, and another about 15% are at risk. But again, I want to highlight that MNDU3 promoter, and that's the same MNDU3 promoter which was used in the Anderson study. So obviously this MNDU3 promoter is a problem. We can't be using this in further clinical trials. So that led us to our central question. Can we generate a UBE3A expressing lentiviral vector with similar, if not improved characteristics as the MNDU3 control vector using a different promoter? Basically, can we change the promoter but keep all the good things about the lentivirus that showed correction in a mouse model? And so I'm not going to show that data, but I'll summarize to say we came up with these two different vectors. So on the top two vectors are the MNDU3 promoters. So these, again, are very strong promoters, but they have the concerns with the oncogenesis and the cancer in the patients. These two vectors are just as strong, they function just as well as these MND promoters, but we have a lot more confidence in their clinical safety profile. You might ask why. So we base our confidence on these results. And so on the left here, this is an article from my lab with Dr. Donald Cohn, where we have a vector that has that same EFS promoter that we designed in our new vector. And so some of the stats from this trial, there are 50 patients, 100% of those patients have survived, 96% show correction of the disease, and there's no incidence of cancer. On the right here is the human PGK, so this is the other promoter. And this is from another clinical trial for a different disorder. There are 30 patients, 90% survival, and none of those deaths are related to the therapy. They're related to the disease. No incidence of cancer and 95% disease improvement. And so from the literature I could find, there have been over 100 patients treated with an EFS vector, not a single incidence of cancer. And there have been over 75 patients that have been treated with the human PGK vector on the right, and no incidence of cancer. So I want to take the time to emphasize lentiviruses aren't dangerous, MND is dangerous. Lentiviruses are a great option to treat therapies as long as you're using them carefully and safely. So this led us to our next question. Do our UBE3A vectors show disease correction of Angelman syndrome to levels similar as the MND vectors? So we were able to show in cell lines they look very good, but what does it look like in a mouse model? So can we show that we can correct Angelman syndrome in a mouse model using these lentiviral vectors? And so this led to these studies in a mouse transplant. So what we do is we sort of try to mimic the gene therapy course that we would see in patients in the mouse model. So we harvest bone marrow cells from the mouse. We give them the lentiviral vector, either the EFS or PGK vector. We transplant those edited cells into recipient Angelman syndrome mice. Then we look at behavioral assays to show correction in behavioral phenotypes, as well as electrophysiology, looking at the EEG signal and delta power specifically, as well as UB3A expression across the various tissues. And so a little bit about what this experimental timeline looks like. 2 days prior to transplant, we harvest stem cells from the Angelman syndrome mice. We transduce them with our lentiviral vectors, so that's again either the EFS or PGK vectors. We transplant about 1 million cells into the Angelman syndrome mouse. And then at week 8, we collect peripheral blood just for early indications if our therapy is working. But at week 14, we start behavioral assays, which I'll show. Week 16, we look at electrophysiology or EEG analysis, which I'll also show. And then week 18, we harvested the spleen, the bone marrow, the blood, and the brain to look at UB3A expression to make sure that the UB3A is getting to all the tissues we hope it would, specifically the brain. And so just before I get into the actual data, I want to go through the conditions quickly. So the top condition here on the top bar is EFS. So again, these are donor male Angelman syndrome cells transduced with our EFS vector, transplanted into recipient Het2 or Angelman syndrome female mice. The human PGK condition is the same, except instead of the EFS vector, it's the PGK vector. And then we have the Angelman syndrome mouse model. So this is the Angelman syndrome mouse, no modifications. And then we have a wild type mouse model, which is a wild type female mouse, no modifications. So think of the red and green conditions as our edited controls. So these are the vectors, or these are edited conditions. And then the gray condition is our negative control, the Angelman mouse, the purple condition being our wild type mouse. And one thing I want to note here is that the recipient mice are 6 to 8 weeks old, which in mouse years is not relevant, but in human years, that's about 14 to 18 years old. So we're transplanting 14 to 18-year-old, the equivalent of 14 to 18-year-old Angelman patients with these Angelman mice. So that's important when we look at our readouts because we're transplanting young adults, the equivalent of young adults in human years. And one thing I want to note is that the colors of the conditions will stay the same. So if you can just remember, red and blue are our edited conditions, gray is our negative control, and purple is our positive control, it'll make these graphs a lot simpler. So again, we wanted to look at behavioral phenotypes. So this is an open field assay looking at distance traveled. They're grouped into different 5-minute bins, but if we just focus on the bin all the way to the right, looking at 25 to 30 minutes, you can see this is looking at distance traveled. So our EFS and PGK mice in blue and red are able to travel to levels the same amount as the wild type, but significantly more than that of the Het2 model, so showing improvement in just locomotive movement. Looking at novel object recognition, we can see that our EFS and PGK conditions on the left show more interaction with the novel object. So this is a readout of cognition. I'm sorry I'm going through this quickly, but just for time's sake. We also did a rotarod assay. So the way this works is you place mice on a rod, and that rod will consistently increase over time. And so we want to see how long it takes the mouse to fall off the beam. The idea being if they have an improvement in motor function, they should be able to stay on the rod for longer. So we do this over 3 trials over the course of 3 days. So the 2 things I want to point out here is that you can see that our EFS and PGK in blue and red are staying on the rod for as long as our wild type mice. And also they're getting better from day 1 to day 2 to day 3. So this is showing not only an improvement in motor function, but also an improvement in learning in that they're getting better at the assay from day 1 to day 2 to day 3, which was encouraging. The last behavioral data I wanted to show was a balance beam. This one's pretty simple. You just place the mice on a beam and just measure the time it takes them to cross that beam. So we have a wide beam on the left, a medium beam in the middle, and then a narrow beam on the right. And you can see at the narrow beam is when we see differences in the time it takes them to cross this beam. And that can be visualized better through these videos. So on the top left, we have our wild type mouse. On the top right, we have our Angelman syndrome mouse. On the bottom left is our EFS mouse, and the bottom right is our PGK mouse. And so these mice are going to cross the beam from right to left, starting at the same time. So you can see there's no difference. So I want to point out that the only difference between the Angelman syndrome Het2 mouse and then our EFS and PGK mouse is that they got those stem cells. So you can see the wild type EFS and PGK mice have already crossed the beam about 10 seconds ago, whereas when we look at our Angelman syndrome mouse in the top right, she's still trying to cross the beam, still trying to cross the beam. So this is just showing an improvement in motor function, and she actually isn't able to cross the beam. She's hanging on, and eventually she falls off. So this is just showing an improvement in motor function, so these mice are able to cross the beam faster. But as I mentioned, we want to make sure that the UB3A is being expressed in the brain. So as we harvested the brains, we did imaging, and so the UB3A is in green, neurons are in red, and immune cells are in orange. So when we look at the staining, you can see the top condition, which is our Angelman syndrome mouse. There's no UBE3A in the brain, which we'd expect. In the wild-type mouse, we see nice spread of UBE3A expression in green. And then in the EFS and PGK conditions, we can see a restoration of that UBE3A expression. But again, this is a cross-corrective strategy, so we need to make sure that the UBE3A is not only in our immune cells, but also in our neurons, right? Because the idea is that our immune cells are going to express UBE3A, which can be uptaken by the neurons. So we need to confirm that. So when we zoom in on these images a little bit closer, again, the left column being neurons, the column to the right being immune cells in orange, and then UBE3A in green. When we look at the far right column, which is the merge, so the overlay of all those three colors, you can see that the green is overlapping with the red, but the green is also overlapping with the orange. So this is showing that not only is UBE3A in our immune cells, it's also in our neurons, which was really encouraging to see. So this is proving that our cross-correction is working. If you can see sort of overlap, or if you see these yellow cells looking, if you look on the right column, if you see those yellow looking cells, that's where green and red are overlapping. So we're showing that the UBE3A is actually present in the neurons as well as the immune cells. And the last bit of data we wanted to look at was, again, electrophysiology or EEG. As we know, delta power is a key readout and sort of the best quantitative readout we have for Angelman syndrome. And so if we look at the spectral graph on the left, you can see that our Angelman syndrome Het2 mice in gray have the high delta power, with our EFS and PGK conditions coming in somewhere in between, with the wild type having low delta power. But when we look on the right, if we just look at the delta power on the figure on the right, you can see that our Angelman syndrome, our EFS and PGK conditions have a significant reduction in the delta power relative to the Angelman syndrome mouse model in gray, and not significantly different than the wild type mouse. So this was showing that not only are we seeing improvement in behavioral phenotypes, we're also seeing improvement in delta power as well. So with that, I hope I've been able to show that we've been able to design a gene therapy gene therapy strategy for Angelman syndrome using lentiviral vectors. In a murine model, again, with young adult mice about 14 to 18 years old, the equivalent of 14 to 18-year-old in humans, we were able to show improvement in behavioral phenotypes, the novel object recognition, the open field assay, the rotarod, and the balance beam. We were able to show brain-wide presence of UBE3A in immune cells and neurons, proving our cross-correction strategy. Improved electrophysiology as proven by decreased delta power. And so for further directions, we need to further characterize these vectors as we move towards FDA studies. For our murine vectors, we need to, again, confirm that this UBE3A is bioactive, and also quantify the UBE3A expression in the brain. I just showed that UBE3A is there. We want to see how much is there, how much of it's in neurons, how much of it's in immune cells. And then for our human vectors, we're doing sort of a lot of the same studies. But one thing we need to do is validate the safety profile of our vectors to ensure that we don't have any incidents like the MND vectors, which we don't anticipate, but we need to prove that as well. So we're working on that at the same time. And so with that, I'd like to thank my lab, specifically my mentor, Donald Cohn, for his mentorship, my thesis committee, my funding, and then the Foundation for Angelman Syndrome Therapeutics, specifically Allison and Jennifer, for trusting and believing in us to bring this vision to life. And lastly, a huge thank you to you all, the patients and families, for your bravery and inspiration. You inspire us to continue to work as hard as we can and as quickly as we can to develop the best therapy we can that will influence and impact meaningful change in your lives. And thank you for your attention.
We have some time for a few questions if anyone has questions for Chris.
I don't see.
We have a question over here.
Allison,
Sue,
Sue again. In the beginning of the presentation, you said that lentiviral vectors have been in 55 gene therapy trials, 406 patients. And then you talk about the toxicity and that kind of thing. In short, it sounds like this has been done before with other rare diseases. And has it been successful? I guess that's my question. It sounds very exciting and we're all on board. Has it worked with other rare diseases?
Yeah, that's a great question. So I had some slides here, so maybe I didn't make it clear, but there have been, of those 406 patients that have received gene therapy, a lot of them have been positive. And the two studies I showed with the side-by-side with the EFS on the left and PGK on the right, those are actual clinical trials for patients that receive gene therapy. So that 97% correction is 90% of patients were cured of the disease for the EFS trial, and 95% were cured of the disease for the PGK trial. And that's just two trials. There's, as I mentioned, 55 gene therapy trials. A lot of them have been successful. I would say not all of them, but a majority of them have been successful, specifically with lentiviruses. There's a really positive, effective clinical track record of these lentiviral gene therapies, and that's sort of what my lab focuses on, and that's what gives us confidence that this would be successful for Angelman syndrome patients as well.
We have one question here.
Yeah, I had a question about, are these immunodeficient mice? Do you make them immunodeficient before you inject? Would that be a problem when you treat humans then?
So yeah, we do condition the mice with busulfan, and the patients would receive busulfan conditioning as well, which is sort of the standard regimen for these patients. Yeah, so the busulfan conditioning is all calculated very closely to know the exact amount to give to get the depletion of the immune cells to make space for the new immune cells to engraft. Yeah, and the mice receive the same conditioning. I just sort of skipped over that. But yes, you're right. Yeah.
Hi, this is Terry Jo Bichelle from Combined Brain. Would you envision that this could be a treatment for every single type of Angelman mutation except for maybe any rare dominant negative?
Yeah, I do believe so, because it's the simple strategy. It doesn't matter the sort of genetic basis of the mutation. We're just overexpressing the protein. So the only key is that there's no UB3A expression. But in the case of the dominant negative, it would probably be a little bit more tricky, and that would have to be a sort of different strategy, which we could probably design one for. But our strategy should be successful for everything apart from that: deletions, insertions, things like that, in that 15th chromosome.
Hi, here. Do you have kids?
No, I don't.
Do you have siblings?
I have a sister, yeah.
Do you like her?
That's my best friend, yeah.
Okay. Would you put this medicine in your sister?
Absolutely.
Okay.
Yeah, I would give it to my 1-year-old niece, whose birthday was yesterday. So yeah, I feel very confident in it, yes.
Okay.
Okay, thank you.
Yes.
Thank you.